My guest today is Professor Yuriy Kirichok, who is Professor of Physiology at the University of California, San Francisco. His lab studies mitochondria, the powerhouse of the cell. Welcome, Yuriy.
Thank you, Gill. Thank you very much for having me.
Yeah, thanks for doing this. So we have a couple of technical papers we want to discuss, but before we get into it, I want to sort of set the context. So your lab is doing a lot of work in this area of mitochondria. Could you describe what mitochondria is in the cell and what it does?
Well, yes, mitochondria are little organelles inside the cell, and they're responsible, for energy production. So they supply cell, with pretty much all the energy it has. So, and the way how it works, our body or in our cells, they kind of, like a special type of, I would say, automobile or something like that. Yeah. And they run only on very high kind of, on fuel of the highest purity, I would say. Okay. So how you can imagine, and only one. Okay, so it's not that it can accept the different types of fuel. Okay, like a premium gasoline or something like that. So, and mitochondria, what it does, when we eat all these different types of food, okay, so it takes all that food and has to convert this into this type, very specific type of gasoline that our body consumes, right? So, and this is what mitochondria does. It just takes the energy out of food and converts it into the energy of a special molecule, which called adenosine triphosphate. So, and this is the gasoline that our body runs on.
So it's part of the cell, and it is really sort of producing the energy needed for the cell to function.
Yeah.
And it's doing so by really taking the nutrients from the bloodstream and then converting that into something that the cell can utilize?
Yes, exactly, because these nutrients from the bloodstream cannot actually fuel the cell. Yes, so all kind of reactions in our body, they all run on a single type of fuel, ATP or adenosine triphosphate. So, and this is what mitochondria have to produce, from those nutrients, from the bloodstream. Yeah.
So is this present in all types of animals, all cells?
Yes, in eukaryotic cells, it's present, yes. Like bacteria, which do not have mitochondria, they have, a mechanism, basically, which kind of, bacteria actually, they are relatives of mitochondria or mitochondrial relatives of bacteria. So bacteria, which do not have mitochondria, they sort of, run like mitochondria themselves. They can actually take, all these nutrients, from the environment and convert them into a single source of energy, ATP, right? So, but for cells, what at some particular, so people think that, very long time ago, like years and years, or billions of years ago, so we a type of bacteria has been engulfed, was engulfed, by a cell, eukaryotic cell, and this eukaryotic cell started to use, this bacteria to produce energy. Okay, produce ATP. Yeah. So that's how bacteria do not have mitochondria; they just do it themselves.
Yeah. So the single cell organisms didn't have it. So evolutionarily, this is a big innovation that allowed more complex multicellular organisms to happen, right? So in some sense.
Some organisms with just one cell, they still have mitochondria sometimes. Yeah. So it's not, that this transition kind of necessarily implies, that organism has to be multicellular. I think, it's overall kind of, maybe an adaptation which allowed, to process, more type of, I don't know, it's just maybe specialization or something like that within the organelles. It's sometimes it's difficult to kind of understand what kind of advantage that would have for monocellular organism, but it does for certain organisms. I think it's just adaptation to a special environment where those cells were or live, right? Which kind of made sense for them to take up mitochondria inside and live with them.
And so in human cells, I would imagine different parts of the body, different cells and different organs and different parts of the body, they all have different concentrations or different numbers of mitochondria?
Yes, right. So heart, for example, has a very high amount of mitochondria. It's about almost like 40% of the volume of the cell. But other cells, skeletal muscle has only about 5%. Yeah, so, and it varies significantly. In addition to that, mitochondria from different tissues are not identical. They have certain differences between them, which makes them suitable to serve a particular type of cell.
And so I guess it's a function of how much energy the cell is demanding, right? So it's almost like for certain cells, you need the big electricity lines, and for others, you can run with a battery and so on. So it's sort of the function of the cell and the energy demand, I would think, right?
Yes, right. And in addition to that, mitochondria, they also source of different metabolites, which might be needed for some other purpose. So, like for example, steroid hormones are generated initially by mitochondria. So it also depends if this particular cell type needs particular type of metabolite which is produced by mitochondria. So mitochondria in kind of energy production is a primary function of mitochondria, but they also a very rich source of different metabolic substrates. Okay, so which certain cell types may need more than others. So, and that's also kind of may dictate what kind of mitochondria are inside a particular cell type.
Right. So this is a very important aspect, and so if it is not working properly, I would imagine all sorts of different types of diseases could happen, right?
Well, yes, definitely. I mean, we have many mitochondrial disorders that has been identified. So mutations, those are usually mutations in certain mitochondrial proteins. And it's very interesting that so the whole mitochondria have about 2,000 proteins in them. Okay, so and among those 2,000 proteins, only few actually kind of produced in mitochondria. So mitochondria usually depends on production of the proteins which are required for the activity on the cell actually. Okay, so and mutations can be either in cellular DNA or in mitochondrial DNA. So a lot of mitochondrial kind of different diseases caused by mutations in mitochondrial DNA. But as much you can say about mutations in the nuclear DNA of the cell as well, which cause mitochondrial dysfunction. So and those are very well known, and some of them are very heavy.
So mitochondria have their own DNA. And so if you look inside the cell, the different mitochondrium that we will see, they all have different types of DNA or are all the same?
No, they have the same. So and we inherit this from our mother. So it's very interesting. So it doesn't come from fathers. It comes from the mother basically. And just because so sperm mitochondrial DNA gets destructed basically after fertilization, and the organism, developing organism only inherits mitochondria from the egg. So this is what happens. So it's very interesting.
Yeah. I remember National Geographic had a program. I think it is sunset now. That if you send a sample of your saliva, they can look at the migratory paths that both your father and mother separately. And on the mother side, because it is mitochondria is coming only from the mother, they could actually look at again disease progression on the mother side to look at progression from all the way, so they can trace it back all the way to Africa 50,000 years ago.
Exactly
and
right.
Which clans you were part of.
That's very interesting. Yeah, I know. This is fascinating. Yeah.
So, so the mechanisms inside the mitochondria. So you mentioned ADP. So this is a chemical that.
So ADP. This is adenosine diphosphate, and this is a molecule which actually sort of which is produced by the body after it consumes the energy of ATP. So we have adenosine triphosphate, which is actual fuel, and ADP this is, what basically what is produced, as a molecule of ATP get used up, basically. And then ADP is imported into mitochondria to attach, the third phosphate, basically, and make, this again ATP. So, mitochondria, they continuously convert ADP into ATP, okay, and deliver ATP into cytosol.
So ATP is sort of the energy storage, right? So mitochondria produces ATP. Let me know if this is right, if I understand this correctly. And when the energy is used, you have ADP that can then be converted back into ATP at a later time by using more energy, right?
Right, right.
And so, I mean, it's so I know there's some history around. Do we really know how the mitochondrium started all the way back in the evolutionary cycle?
Yes, right. They were bacteria at some particular moment, and bacteria, they actually have mechanisms for energy production which is reminiscent of mitochondrial energy production. They have even some similarities between specific proteins within them, bacteria and mitochondria. But bacteria kind of look much more simple, okay? So they obviously yeah. So bacteria, well, it's just okay. Mitochondria, they have like this kind of proteins which are responsible for energy production within mitochondria. Let's say, like, I'll give you one example. For example, complex 1 of the electron transport chain, which is one of the kind of major molecules which are responsible for energy production in mitochondria and bacteria. So, in my mitochondria has more than 40 different subunits within that complex, but bacteria has only 16. Okay, so bacteria kind of, they are less sophisticated example of like energy production machinery, or like this kind of power plant, I would say, than our mitochondria. Because in our mitochondria, the cell provides additional proteins to mitochondria and modifies, basically, which insert themselves into this machinery for energy production, incorporate them, and perhaps impart certain types of specific regulation, cell-specific regulation, and maybe more complexity, so that our energy production machinery basically can respond to particular needs of our body and our cells. Okay, so that's how, like, yes. So it's just basically a more sophisticated energy production machinery.
Right, right. I know that we're going to talk about this a little later in the paper. So again, mechanistically, when this ATP production happens, there is also heat being generated. What is sort of the mechanism there?
Yes, so that's kind of interesting. So mitochondria, they do not only produce ATP. I told you that they only produce ATP previously, and this is what people kind of primarily focus on. But energy-wise, they produce actually two forms of energy: ATP, which serves as a fuel for our body, and heat, which makes us warm. Okay, so helps basically to support body temperature. So, and this is basically what mitochondria are responsible for. And some mitochondria, they produce primarily ATP, the fuel, like, for example, those mitochondria in heart and skeletal muscle. But some mitochondria, they focus on heat production. We have a specialized tissue in our body which is called brown fat. The brown fat is specialized organ which is kind of heats up our blood, in essence. Okay, so blood vessels go through that organ, and it generates heat by itself, and heats blood. So, and then the blood goes throughout our body and kind of maintains the body temperature. So brown fat mitochondria, they specifically designed, they specifically work to produce heat, not ATP. They produce just a little bit of ATP and primarily heat. So in principle, you wouldn't even say that, you couldn't get, this general statement that mitochondria are primarily exist, for heat, for ATP production, because, some of them actually are so different, from others that they focus on heat production.
Mm-hmm. And that's a critical function too. We have to keep the body at some specific temperature.
Right.
And so you have a paper here, Yuriy. So mechanism of fatty acid dependent UCP1 uncoupling in brown fat mitochondria. So you mentioned brown fat mitochondria. So what exactly is the UCP1 uncoupling that happens?
Uncoupling, what it uncouples? It uncouples burning of food from ATP production. So what it does, it actually, in essence, so normally mitochondria would produce ATP from food, right? So, but UCP1 uncouples that connection and makes, food to be converted into heat instead of ATP. Okay, so this is what it does. So it's, that's how it's called uncoupling protein. Okay, because it's uncoupling between food oxidation or food burning and production of ATP. Okay. So, but mechanistically, what it does, it introduces a particular type of current, because in mitochondria they run on electricity. So, and this uncoupling protein 1, it introduces a particular type of proton current into the circuitry of mitochondria, which is responsible for energy production, as if like it short circuits mitochondria in a way and make mitochondria produce heat.
So. So can you take any mitochondrium and essentially make it produce heat rather than ATP?
Yes, right. So this is the case as well, because brown fat mitochondria kind of channels, pretty much 100% of food oxidation, energy of food oxidation into heat. Other mitochondria, like heart mitochondria and skeletal muscle mitochondria, they channel primarily all energy normally into ATP production, but not all energy. About 20% of energy in skeletal muscle, for example, is actually goes to heat production. Okay, so and we can in principle make it even more. So we can sort of because in essence skeletal muscle mitochondria can produce much more than it produces at rest when we sit. Okay, when we run it can produce 10 times more energy. Okay, so this is how much the energy kind of output of mitochondria can change. And in principle we can increase heat production by the skeletal muscle mitochondria when we sit without even disruption of ATP production, because mitochondrial energy sort of capacity would be able to accommodate both loads, right? And we can make skeletal muscle mitochondria producing more heat and still produce ATP for whatever we need. So, and this is a way to lose energy or burn fat, for example, because we just increase energy production and dissipate heat into environment, like burn calories. This is how kind of the simplest way to explain this.
So because the body has to keep, I don't know much about this, Yuriy, I'm just just asking, because the body has to keep at some specific temperature, if mitochondria start to produce a lot of heat, then the body has to take evasive action to get the heat out of the system, right?
Yeah, and that's definitely mitochondrial heat production is not the only mechanism of regulation of body temperature. So it has kind of, the system is a little bit more complex than that. So mitochondria produce heat, but our skin constantly dissipate, kind of gives out that heat into the environment. So, and by increasing or dilating blood vessels in the skin, we can actually dissipate more heat. So what normally happens, when mitochondria increases heat production, the blood vessels will dilate and we dissipate more as well. So it's kind of, it's mechanism which controls, both production of heat and also dissipation of heat, so the body temperature stays constant and everything is controlled by brain. It measures temperatures everywhere and makes sure that, everything kind of, the whole system sort of, works, as a whole, coherently and harmoniously.
Yeah. And so E is E equal to MC squared. So if the body can create heat and dissipate heat, this could be used to reduce weight.
Weight, yes, exactly. That's exactly true. So it's not that we necessarily need kind of this the theory of relativity to understand everything about heat production in mitochondria, but you are in essence right.
And so I know that your lab is working on this idea. So obviously we have a big problem in the US and most of the Western countries with obesity Generally speaking, obesity leads to a whole sort of metabolic diseases, diabetes, hypertension, and so on. So is this a mechanism we can use to tackle obesity?
Yes, totally. So what basically, if you take a look at the current approach, how people trying to control weight, everybody right away, would think about diet and exercise. But, a heat dissipation is, normally is a third way to lose weight. So, and it has been used, by our ancestors, on a regular basis, as they were living in caves and we were kind of, cold and we were sort of burning a lot of food, just to heat themselves up and keep body temperature at 37 degrees. So now when we are exposed, to this comfortable living temperatures, we're using this mechanism much less. So, and in principle, we would perhaps want to compensate for that. And in addition to dieting and exercise, it would be great to develop, some sort of, therapeutic intervention which would help us, to increase heat production by our bodies as it was in the past. So that would help, to lose weight. And perhaps, can be used alone, just by itself, or in combination with dieting and exercise, and would increase kind of, capacity of our body to stay within a certain weight range. Because, usually the problem is, with dieting and exercise, you lose weight and then you gain it again, you lose it, gain it again. So I hope, that when we introduce, the third missing component, we would be able to achieve more sustained and more profound weight loss for many people.
Yeah, it's very attractive. I don't know the metrics, Yuri. My understanding is that it's really difficult to lose weight by exercising because the amount of energy that you're spending in the grand scheme of things is not as high. Dieting is more effective, but this mechanism is potentially much more efficient, right? If you can essentially radiate heat away, you could potentially lose. No, it's so attractive to say, take something, go to bed, and when you wake up, it'll be 5 pounds lighter. That would be that would be not more attractive to most people.
Well, I would just say that, it's not more efficient than exercise. So with exercise, you normally would achieve, higher energy expenditure, but people cannot exercise for the whole day, right? You usually can devote to exercise, maybe 1 hour, 2 hours per day, right? Maximal that normally people would do. But this mechanism, heat production, you basically can use all the time. And the beauty of it, that you can increase energy expenditure by heat production just a little bit, but because it goes on the ground all the time and dissipates heat and burns calories, so it just kind of, eventually it achieves, the same efficiency or maybe more efficiency than exercise. So that's basically the difference. Other than that, actually exercise and heat production, they're using almost the same metabolic pathways to burn food, to burn calories. Almost the same, almost identical. So everything kind of, upstream of mitochondria is the same for exercise and heat production. But, we can use heat production kind of, as we go about our normal routine, and it just does it on the background. For exercise, you have to actually focus, on exercise.
Right. And so would we see an appreciable difference? Let's say you set your thermostat at 70 typically, rather than that you set it at 60 when you go to sleep. Would we see an appreciable difference in sort of energy consumption?
So most likely not, because what happens when people set the thermostat lower temperature, they put more kind of clothes and like blankets and everything on themselves. So so this is because this is kind of another way to preserve because it doesn't feel comfortable. Okay, so and for our ancestors, it didn't feel comfortable living at colder temperatures. You actually have to go through the process of adaptation. So and you can be adapted, living at colder temperatures, but nobody would normally venture into this. So when you cold adapt, our body kind of increases the size of brown fat. Okay, so and does some other adaptations which sort of helps to produce more heat. But it takes a month or something like that, to fully adapt. And while you're doing this, you'll feel uncomfortable. Okay, so and eventually, like it's not very practical. You wouldn't be kind of showing up like without dress, for important meetings and stuff like that. So it's all the kind of complications which involved in that, which prevents just kind of dialing down the time thermostat, and just kind of losing the same amount of heat. So it's difficult. Yeah, more difficult than that.
I know that your lab is working on. So can we intervene therapeutically to increase the heat production in the mitochondria?
Yeah, so for that, we need to control these proteins, in mitochondria, which are responsible for heat production. So these proteins, because what happens, mitochondria run on electricity. So, and the first step of energy conversion in mitochondria is actually taking food, burning food slowly, and converting food into voltage. So across the inner mitochondrial membrane. Okay, they have two membranes, and there is a voltage which is generated across one of the kind of internal kind of membrane, which is sort of inside the mitochondria. So, and after that, that voltage inside mitochondria is converted to ATP and heat. Okay, so the protein which produces heat simply short circuits that voltage and make it produce heat, basically completely kind of dissipate that voltage and makes it produce heat. So there is a special protein for that. So if you can control kind of proton current, right, which short circuits that voltage, mitochondrial voltage, with some sort of drug, so that would do the same. So, and unfortunately, like nothing like that was available. So we knew like how induce this proton current by completely artificial way, and actually that was one of the kind of one of the perhaps most known anti-obesity medication in the whole history of mankind, which is called 2,4-dimetrophenol, which is dangerous to use actually. Because it introduced this proton weak in a completely artificial manner in mitochondria, bypassing the native thermogenic pathways. Okay, so but what should be done? You should control native thermogenic pathways, native proteins which are responsible for proton leak in mitochondria. Okay, so and that should be developed. So and because we couldn't understand really all this mechanism which is involved in that proton leak and how all these proteins work and even their identity. So that's how it was difficult to control native thermogenic pathways. In my lab, I'm working on understanding like what proteins are involved in mitochondrial proton leak, which short circuits voltage, and how to control them therapeutically, pharmacologically. So and I hope that in the future we'll be able to achieve a very mild, safe stimulation of mitochondrial thermogenesis, which will be helpful for people to lose weight.
Yeah, that's really fascinating. So if I understand this correctly, Yuriy, so it's the electric voltage of the mitochondria that sets it into two different modalities, the ATP production and the heat production. So if you have a dial on the mitochondrium and you can sort of turn it one way or the other, it could behave either way, right? If that's possible.
Yeah, right. Yeah, you can think about that, mitochondria is about like a power plant, electrical power plant, which generates electricity. So you can just kind of abstract yourself, from all this complexity of like cell and mitochondria and something like that, and just kind of, take a kind of more general and more simplified view. So mitochondria burn food, which is like carbohydrates, and other stuff, like a fuel basically, and it generates voltage in a very similar way to the power plant, which burns, for example, coal or something like that, and generates voltage out of it, right? So, and then, like, imagine it kind of, has like this electrical outlet where you can plug different devices. One device, can produce work, right? So, and makes us kind of, move kind of, our brain work, our muscles contract and something like that. And another device, just like a heater, which produces heat and nothing else. So that's how, like, what's the difference between ATP production and heat production. We just kind of, plug different devices into the same electrical outlet.
Yeah, so is it possible, I don't know if this is true, when you sleep, when you're not eating, is the ATP production lot lower?
No, it's not, because we actually, so the thing is, we always have fat, right? And we also have, storage of other nutrients in our body. But fat, is one of the examples, right? So, it can actually keep you going, like, for like a month or something like that. For some people, it might be like a year, if you can see the amount of fat, and some people accumulate. Fat is used like an energy storage, which can be kind of, used by mitochondria gradually. So, and although like, the fat is stored, in some particular places, right? So it is hydrolyzed basically by the cells, which store fat. And then like, these fatty acids, they release in the bloodstream and they go everywhere in our body, reach heart, reach skeletal muscle, brown fat, everything. So that's how like, this fat, it's just like days and days of energy. Doesn't matter where we sleep, or we eat, and things like that. So we have it, in our cells, in sufficient amount. So energy, well, ATP production doesn't necessarily go down, when we sleep.
So this therapeutic mechanism that you're working on. So if you go inside a cell, let's say there are 100 mitochondria in there, can you selectively? How much would be there? Thousands. But can you selectively take some of them and turn them into more heat-producing things?
Well, perhaps that would be difficult. So I would say perhaps pharmacological. Well, a drug can be developed, which specifically targets particular organs. That would be more easier. Like for example, skeletal muscle usually idles. So and so skeletal muscle perhaps can be used very efficiently for heat production too. So and so it's much easier actually to target a particular tissue pharmacologically than kind of a subset of mitochondria within a particular cell.
I don't know if I understand this, Yuriy. So for the mitochondria to produce ATP, it needs ADP and the phosphate as sort of raw materials, right? So if those things are not available, then ATP production will slow, I would think, right? So, so what are the implications of that? Is it possible for us to sort of remove ADP and phosphate and put that into heat producing mode?
No, it wouldn't produce heat because it will not increase, that proton current. Because that proton current, through a specialized proteins, within mitochondria, which short-circuit voltage, those proteins, they have to be activated themselves. That proton link has to be normally, without long-chain fatty acids, for example, which are native activators of mitochondrial thermogenesis, which sort of, act upon those proteins, and induce proton current through them. Without fatty acids, you wouldn't have any current, like zero, through those proteins. And altering the amount of ADP available won't change that as well. So we actually need to provide actual activator. So if you limit ATP production, it will not be good because you obviously may end up with the energy deficit in the cell and kind of like general damage for the entire body. So that's how the best approach is to leave ATP production alone, okay? So and just let it go and control heat production separately. And do not kind of activate heat production too much as well, because if you do, then you can reduce that voltage, mitochondrial voltage substantially, so that ATP production slows as well. But in some kind of boundaries, within certain boundaries, we can plug into the mitochondrial voltage, kind of these two appliances, and it will be fine. It should be fine because mitochondria are pretty powerful. So normally, as we see it right now, so we consume 10 times less energy that mitochondria are capable of producing. Okay, so that's how there is some limit, where we can sort of increase, kind of put more load into mitochondria and they will be fine. But we shouldn't overload them with heat production.
So there's a lot of capacity there. So, so if you're successful with this sort of therapeutic intervention for, let's say, obesity, would that be like a tablet or some injection?
Orally, yes, bioavailable medication, basically that you take, and and you just basically have to take it maybe like once per day or 2 times per day, something like that, or whenever you want to. So and that would be it. So and you just kind of increase heat production just a little bit, so you don't really feel any increase in heat production really, or like increase in body temperature, because skin can still compensate and just lose it right into the environment. So but that would go on the background like for the 24 hours, and it would burn eventually a significant amount of calories. As a matter of fact, although mitochondria is capable to increase overall kind of energy production 10 times, it would only increase heat production like that, by this medication, maybe only 1.25 times, okay, only by 25%. And that would be sufficient to burn approximately 1 pound per week, just like that, like on the background, without even thinking about that or feeling much.
The weight reduction drugs haven't had a great track history. Do you see, what do you see from side effect perspective? Do you see any issues?
Well, yeah. So from the point of side effects, we shouldn't overdo it, definitely, because you can think about increasing heat production as about stimulation of the same pathway as exercise does, the same metabolic pathways. Okay, so because those two kind of because again, you basically you're plugging in your heat production in the same outlet as kind of exercise, right? So and for that reason you can always think about, so you get exhausted when you exercise for a very long time, very intensively, right? So one of the biggest perhaps problems not to overstimulate, not to put too much load on mitochondria, but overall we expect that effects of increase of heat production would be maybe even they can bring even more benefits, because they would replicate to some degree beneficial effects of exercise on our metabolism in general, kind of maybe upregulating all this kind of pathways for energy production in the body, and which would make us sort of having stronger metabolism, more powerful metabolism, and things like that. Maybe reduction of oxidative stress as well, because exercise does that. Exercise has many different kind of beneficial effects. Perhaps when you talk about kind of how to reduce the aging of the body, exercise one of the best things you can do for yourself. So we actually hope that maybe, we have to see, but we hope that maybe increasing heat production might have some of the beneficial effects of exercise, but we shouldn't overdo it, for sure. So this is because too much exercise is bad for you, as you know. So you shouldn't over kind of exert yourself. So the same thing with heat production. So we need to be careful. We need the drug that has to be developed, it has to be such that it would be self-limiting. Okay. So when people kind of go and accidentally take more than needed, it doesn't result in adverse effects. Yeah.
Yeah, I guess that would be the challenge. If the product is really effective, and people want to reduce weight very fast, that may not be the right thing to do, right? You have to really take it gradually, I would think.
Exactly right. But I think from what we currently know about the mechanism, how it could be activated, this proton current, these proteins which are responsible for heat production in mitochondria, there are drugs that perhaps can limit themselves, meaning that we can design drug which can will be kept at certain maximal level of proton current which it can induce, and wouldn't induce more even if you increase the concentration higher. Okay, so we sort of can perhaps, I hope, incorporate into the drug itself this sort of like a limit where it wouldn't go above certain level. So plus to that, I really hope that because we stimulate this native pathways for thermogenesis, there are we actually know that there are some mechanisms which tell this heat production machinery that it has to stop because ATP production can be compromised because it dissipates too much voltage. So we see in two of these publications that you mentioned, we describe certain of those mechanisms, like dependence, because the mechanism for heat production can sense, for example, voltage, and if voltage drops, mitochondrial voltage drops significantly, so the proton leak, which dissipates that voltage, is reduced very significantly too, to prevent further dissipation of voltage. So there are certain pathways which we hope will help us to overcome that, but again, this is a technical problem which has to be overcome, and eventually I hope we will come up with something safe and efficient.
Yeah, I mean, part of it is potentially a formulation. So suppose you have some sort of a sustained release formulation that you take once a month or something like that, right? So rather than popping pills on a daily basis.
Yeah. So in principle, it would be great if, for example, we can, it's like with insulin, for example, taking too much insulin would be dangerous, right? So, but there are some pumps which can be the small pumps which can be implanted, which reduce inject just correct amount of insulin and monitor blood sugar level, right? So we might monitor, during such injection, heart rate, for example, because that would be indication that because we expect that heat stimulation like exercise may increase heart rate, and for that reason we can monitor heart rate and body temperature, for example, and as soon as it happens, just injection stops. So, and that might help to control it. But plus to that, as you mentioned, so there are actually drugs which control release, right? Which you take and and then they sort of just give up the active compound slowly. Okay, not you take and the whole thing just goes into your blood, but it just goes slowly, releases a little bit over the day, and then you don't have very significant peaks of the drug in the bloodstream, which help you to prevent the overdose.
Is it possible, Yuriy? So can you go to a specific organ like the liver, and I don't know what you could do, but could you affect the mitochondria in that particular organ in some way to sort of change the behavior?
So in essence, liver is usually the first organ which is exposed to drug, and liver actually decides as well whether to release the drug to a significant degree or just cut it in pieces and just So usually people when develop drugs they take a look at liver very take a very particular look at liver just to make sure that doesn't chop up the drug nothing is left after this in 10 minutes or something like that, and there is no effect at all. So but liver is exposed to drugs right away normally when you take orally. And for that reason, we expect that liver, for example, will be like one of the first targets, of our medication. So, but that said, perhaps, pharmacological approaches can be developed to target, to skeletal muscle, for example. So, there are approaches like that, which can be taken as well. But that said, liver, by itself, the the low-hanging fruit, which we can control mitochondria in right away, perhaps. It's so important, for metabolism that it will be already very important, target tissue. Like for example, fatty liver, right? So people, who accumulate fat in their internal organs and liver, for example, they often also have type 2 diabetes, and fatty liver is one of the complicating factors in the condition, actually. So as soon as we can burn out all the fat from liver with this approach, by increasing heat production by liver mitochondria, we can actually fight fatty liver disease and also, perhaps, reducing the metabolic syndrome to a very significant degree. So I really hope, that it's one of the sort of, low-hanging fruits that we can go after. Yeah.
Yeah, it's really exciting research. It seems like it could have a lot of different beneficial effects all on that metabolic syndrome arena, right?
Right, that's true. Yeah.
And I don't know the exact numbers. 1/3 to 1/2 of the healthcare costs that we have could be attributed to those things like type 2 diabetes, hypertension, and so on. And so any small effect there, still quite billions of dollars from a healthcare perspective.
Yes, but why I'm really excited to work on this project is because the number of potential indications that we can pursue is really immense. So you can see even polycystic ovary syndrome in women and women infertility can potentially be alleviated very significantly with this drug. So this is kind of we're also thinking about this indication in particular, just because it would introduce so much more joy into the whole family life for some people. Yeah, because the same is so the driving factor behind polycystic ovary syndrome is also metabolic. So it's metabolic, so it's insulin resistance primarily. So and many of these patients are obese, majority of them, and they have other kind of metabolic abnormalities. We expect that this drug may help these patients very significantly, and actually perhaps we need to see, but perhaps can restore fertility as well. So that's how you wouldn't think about such a particular application for this technology, but it may exist actually.
Yeah. So I want to finish up with your other paper. H+ transport is an integral function of the mitochondrial ADP, ATP carrier. H+, the hydrogen ion, that is basically proton, right?
Yes, it's proton current. Yes, this the same that proton current that I was talking about, which induces production.
And so what are we finding here? I know that the ADP and the ATP transport is a complex mechanism, but you're finding something slightly different.
Yes, well, it's kind of very interesting. Like when you publish a paper in a scientific journal, it's sometimes for people kind of just because the way how you present this, it's very difficult to understand really kind of the actual importance, like in a nutshell. But the nutshell is very simple. So when you use a protein which is responsible, for heat production in mitochondria, in brown fat, the specialized thermogenic tissue, it was uncoupling protein 1 or UCP1, right? And that was the first paper. We characterized, how fatty acids activate proton current in that protein, and that's how, it was, such an important publication. But the protein which is responsible for mitochondrial heat production in all other tissues like skeletal muscle, liver, heart, everywhere. It was not really known. It was really controversial. Decades of research went into this, just kind of to end up with a bit of controversy about what proteins is responsible. So this paper actually demonstrates for the first time. It reports proton current, which induces heat production in mitochondria directly. So we kind of succeeded in measurement of this tiny electrical currents from a single mitochondrion from the cell. Okay, so and we demonstrated that how we could then see what happens when you eliminate certain proteins with that current. And we demonstrate in this paper that when you remove a protein which is responsible for import of ADP into mitochondria and export of ATP outside of mitochondria, this crucial protein which is important for kind of the whole ATP production machinery. So the same protein actually is responsible for heat production in mitochondria, and it's kind of coordinates in mitochondria of regular tissues like skeletal muscle, not thermogenic tissues, but regular tissues. It coordinates ATP production and heat production very tightly to make sure that everything works harmoniously and we don't produce too much heat to overwhelm ATP production and vice versa. So that was kind of identification of actual protein involved, and also the mechanisms, kind of physiological mechanisms which are involved in heat production in skeletal muscle. So that's kind of despite of sort of very kind of a little bit sort of interesting title, very mechanical title. So this is what the paper is actually about.
So in conclusion, Yuriy, going back to sort of the heat production, turning on heat production, perhaps with a beneficial effect on weight loss and related diseases. If you look forward, how many years do you think it will take for a practical product?
I think so as a matter of fact, we have molecules, we have already activators of these pathways. So the only thing, this is the first, prototypes. Okay, they do activate, this native pathways, and this is the first specific activators, this, native pathways. But they have to be adjusted from the point of view of medicinal chemistry. basically, we need to make sure that they stay in the bloodstream, for, long enough time. And, we need to play, with toxicity because, it's very important, they don't have any other targets and things like that. So the process of selection of the final molecules, because, we will be introducing modifications, to the existing prototype molecules. So the whole process of modification might take us, about, 4 or 5 years before clinical trials, maybe shorter. We'll see. It will depend a lot, on, maybe we'll be lucky, it will be faster. But, my estimation, it might take us, about, few years, basically, not so many. So I definitely think, below 10 to get, to the clinical trials, and actually see what happens, what kind of benefits, we can bring, into the society with this type of drug.
Do you have a preclinical model, like a mouse model or something on this?
Yes, right. So we will be running this on rats. So this is basically right now, we were using cells as our models, until now, and isolated mitochondria. And the mechanism is confirmed on the level of the mitochondria and the cell, whole cell, right? So we are right now graduating to the level of animal models. So, and we'll be running experiments with rats most likely, because, they're larger animals, which, where metabolism is a little bit more like in humans, because, mice, have super fast metabolism, because, they're so small. Anyway, so we'll be running, animal experiments, shortly, toxicity experiments also in animals. And right after that, we perhaps, can kind of graduate, as soon as we show everything, no toxicity and efficiency of the drugs, we will be graduating them, we'll be speaking with government about kind of granting us, permission, to conduct, human trials, but before that, many other experiments has to be performed, to kind of, to satisfy all the equations about safety and lack of toxicity and things like that, before we engage in that. So it takes some time. It takes some time.
How far are you from clinical trials?
From clinical trials, as I said, I think, we might be away, 3 or 4 years, from clinical trials. Yeah.
Yeah, good luck with it. It seems like a very important area, and if you're successful, it will have a big effect.
Bill, thank you very much. I really hope so. It was kind of, one of those, dreams that I had, in the beginning of my scientific career, that, I can, this whole area of heat production really fascinated me. And also because, I'm a physicist by education, the electricity involved in the process also was really attractive, So I really hope, that I can sort of, bring this kind of, my, expertise in physics, to kind of, to bring some progress, into area of energy production in biology as well. So it will be kind of, realization of my dream, as a scientist.
Excellent. Yeah, this has been great, Yuriy. Thanks so much for spending time with me.
Absolutely, my pleasure. It was great questions as well.
Thank you.
Thank you.